Greetings. This is Alberto Villarengo here to present my presentation on glycolysis, gluconeogenesis, and the involvement of aldolase in those processes. A video link will be posted to this very title slide once the presentation is uploaded and complete. So, metabolism. We intake food, we digest it, it yields us energy for us to function as living organisms. We get that energy from glucose. Glucose is a carbohydrate and one of two well-known monosaccharide sugars, the other being fructose. It plays a pivotal role in organic metabolism. Glucose has rich energy potential, can polymerize for storage purposes and it's has metabolic versatility in its in its products that can yield into other metabolic cycles. All this renders it to be the optimal energy source for life on planet Earth. Of all the metabolic processes glucose is involved in, there are two that we should all know about. Any biochemist must know. There's glycolysis, the breakdown of glucose into pyruvate, one glucose into 2-pyruvate, and as well as other metabolites, and gluconeogenesis, which is a conversion of a non-carbohydrate macromolecule, like a lipid or a protein, into glucose. Now, our metabolism in a nutshell consists of we intake oxygen, water, food, and chemical energy, and we output, and as a byproduct, we output carbon dioxide, sweat and breath, urine, poop, and heat, body heat. But right now we're focusing on a little more detailed stuff. So let's focus on, right now we're going on into more detail. This presentation, we will cover glycolysis and gluconeogenesis. Both will have an explanation and diagram slide. We'll also go over the enzyme aldolase, how it's, the purpose and function it serves, as well as the isoforms of aldolase and the transcription factors that determine which cells of ours carry certain aldolase isoforms. All right. Start with glycolysis. Just take my face off of here. So glycolysis, in it basically, glucose, we intake, it oxidizes and it turns into water, pyruvate, ATP, which is an energy yielding molecule, and NADH, an intermediary molecule that helps with other metabolic processes like the electron transport chain, citric acid cycle, and so on. In general, the reaction equation of glucose is you get glucose plus 2 NAD positive ions, 2 ADP molecules, and 2 pi. You get 2 pyruvate molecules, 2 NADH, 2 hydrogen ions, 2 ATP, the energy molecule, and 2 units of water, H2O. Now, some may argue glycolysis only occurs in aerobes, but in truth, it occurs in aerobes and anaerobes. It does not need oxygen for it to happen. Truth be told is that the confusion, though, stems from what happens after glycolysis. You see the product metabolic processes after glycolysis, like citric acid cycle, electric transport chain, and so on, and as well as fermentation. Those occur depending upon whether oxygen is present or not. Fermentation when oxygen is not present, electron transport chain and citric acid cycle when oxygen is present. That's a whole other story, though. Keep that in mind for future lessons. Glycolysis itself uses a whole bunch of different enzymes in the enzymes to make it happen. And it all happens in the cytosol or cytoplasm of the cell. So it kind of just floats around, and these enzymes kind of float around and wait for the intermediaries to bind to them and so on. First, have them all listed here too. Hexokinase, phosphohexose isomerase, phosphofructokinase 1, aldolase, which we'll go into more detail of aldolase later, triose phosphate isomerase, glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, and pyruvate kinase. Big mouthful, but they're all listed here, so you can go back to this presentation and write them down for your notes. And they're all in order too of what they occur in, of the order they occur in glycolysis. This here is a little diagram of these images here are how glycolysis happen. You see, we have the preparatory phase where energy is actually inputted into glycolysis, causing it to which you see, glycolysis doesn't just happen spontaneously. It does need energy input for it to happen. One of those things where we must expend energy to get energy. Start with your glucose, breaks down, the hexokinase breaks it down into glucose phosphate, and which can reverse it, and then so on. Eventually, you get your eventually the glucose breaks into glyceraldehyde phosphate and dihydroxyacetone phosphate, the latter of which is converted into another glyceraldehyde phosphate. Now you have two glyceraldehyde phosphates, and this is where the payoff phase, where the energy yield actually happens. And it goes through the payoff phase, eventually becoming 2-pyruvate, as well as 2 units of ATP. Another couple units, as well as becomes pyruvate and ATP. You get 2-pyruvate, and by the end, should have 4 ATP yielded. Considering it takes 2 ATP to start glucose and 4 ATP yielded from glycolysis, you have a net yield of 2 ATP, not much. Luckily, the electron transport chain produces a whole bunch of ATP. Keep that in mind for future lessons. Now let's go into gluconeogenesis. You see, not all of our energy sources are glucose. However, in order to, for lipids and proteins can be used as energy sources, but before they can be, they need to be converted into glucose. Gluconeogenesis converts these non-carbohydrates into glucose. Careful not to confuse it with glycogenolysis. Little similar sounding terms, but glycogenolysis breaks down into pre-existing carbohydrate glycogen into glucose. Gluconeogenesis is specific for non-carbohydrate molecules turning into glucose. Basically, gluconeogenesis is like a reverse glycolysis process, but before it gets to the reverse glycolysis part, it must convert the non-carbohydrates into something called phosphoenolpyruvate. And this process can vary between different precursors, different non-carb macromolecules. Like lactate and the amino acid alanine must first be converted to pyruvate, then oxaloacetate, and a few other steps before entering reverse glycolysis. I have it all written down here for you guys to write in your notes. Then you have your amino acids, aspartate and glutamate, need to be converted via the triboxylic acid or citric acid cycle into the necessary intermediates before undergoing gluconeogenesis. Glycerol is similar. It gets broken down into through a series of into glyceraldehyde 3-phosphate, and then it enters gluconeogenesis. All right. This here is a little diagram of gluconeogenesis in our human cells. The phosphoenolpyruvate, shorthand PEP, like I said before, must be made first to start making glucose. It takes place in both the mitochondria and the cytosol. Now, remember that long list of the lactate and alanine steps? Well, sometimes a couple steps can be skipped if the because there's like not just, let me backtrack a little bit. You'll see in the lactate and alanine process that it turns into that after that it turns into pyruvate, oxaloacetate, malate, and then oxaloacetate again. That part is what happens if it becomes pyruvate and then leaves, and then has to leave the mitochondria into the cytosol before resuming the process. However, the mitochondria have such enzymes in them too, so not all of the oxaloacetate has to leave the mitochondria as malate before becoming PEP. Sometimes it can become PEP, phosphoenolpyruvate, right off the bat. Now, you may have noticed in the gluconeogenesis, in the glycolysis diagrams, the enzyme aldolase. Now, aldolase is a pretty interesting enzyme. You see, not all the energy sources. Turns out, although glucose is the ideal energy source, it's not the only monosaccharide sugar we can harvest energy from. You see, aldolase has the ability to cleave fructose-based sugars and become fructose sugar-based molecules like fructose biphosphate. It can cleave them into, has the ability to cleave those. As such, it allows us to metabolize both glucose and fructose. There are two major classes of aldolase. Class 1 is found in animals, plants, and green algae. Class 2 is found in bacteria and fungus, like mushrooms and yeast. Now, it's important to aldolase reversibly cleave it. Aldolase doesn't just go one way too; it also can reverse the process, allowing the process to go in reverse. So it plays a role in gluconeogenesis as well. Okay, its main purpose is to break the sugar ring into the 3 carbon units that eventually become pyruvate. And of course, in the reverse, it binds the sugar ring from the precursors that would make pyruvate. All right. Now, aldolase has different isoforms. We're going to focus on human isoforms of aldolase. You see, there's isoform A. This particular aldolase is found in muscles, in your muscle tissues, and it works it metabolizes your fructose. It's more optimized to metabolize your fructose for glycolysis. Isoform B is exclusive to your human liver. It's found in your liver, and it metabolizes your fructose intermediates for gluconeogenesis. You know how like the liver is the organ that sifts through your basically it sifts through whatever nutrients you consume and packages them or stores them as fat for later use. Well, isoform B aldolase does that with fructose and turns it into either glucose or turns it into glucose for immediate harvest or for storing as fat. And then isoform C. Now, isoform C of aldolase, there's still research going on that and what it's meant for. However, It is found in brain neurons, and it's has been linked to signaling pathways for for stressful situations involving low oxygen environments, involving hypoxia. So it could be hypothesized that isoform C of aldolase becomes is meant for catalyzing glycolysis in the brain when hypoxia is starting to kick in, like sort of way to help keep us going when oxygen starts going low. These here are different images of the aldolase isoforms taken from Protein Databank reference. You get different arrangement. You'll notice how in each of these images, they're like different protein strands. Aldolase is like one of those quaternary proteins. It consists of multiple tertiary structures bound together. And you'll notice how each of these different tertiary structures are arranged in different patterns for each isoform's quaternary structure. These simple rearrangements are what optimize the aldolase for the different metabolic processes in the body parts aforementioned. Furthermore, and what tells our different body parts which isoform of aldolase to produce are transcription factors. These transcription factors, they're proteins that bind to portions of DNA, and these proteins, like an enzyme, will either block, like an inhibitor, block RNA polymerase enzymes from attaching to it and transcribing it, or they will, like an enzyme, like a coenzyme themselves, prompt the RNA polymerase enzyme or draw the RNA polymerase enzyme to that specific site and ensure that factor is transcribed sufficiently. Some important transcription factors to go over is especially for aldolase B, for the aldolase B isoform. Your HNF1 is a strong activator of the aldolase B promoter, and HNF3 is an anti-activator. It blocks the transcription. And interesting enough, HNF1 and 3, they compete with each other. It's a matter of which one gets to they compete with each other, trying to get to a single binding site. But depending upon the type of cell specialization, only one will make it. And then the other two transcription factors are C/EBP and DBP. C/EBP, it's a weak activator of aldolase B. It doesn't work as strongly as HNF1. Another interesting thing is, and DBP is a strong activator like HNF1, but it cannot work simultaneously with DBP, with C/EBP. Basically, C/EBP and DBP were both, if they were both cannot both be on the binding site. However, interesting enough, as I mentioned earlier, HNF1 and HNF3 cannot bind at the same time because they compete for the same site. Interesting enough, HNF3 and DBP can simultaneously bind, but they will essentially nullify each other via like a net effect. DBP will activate, HNF3 will anti-activate, and so they basically both bind to the site and activation is canceled out. So, what have we learned today? Well, in summary, we've learned that glycolysis breaks down glucose into pyruvate and other metabolites, while gluconeogenesis takes non-carbohydrates and synthesizes glucose to be broken down again into energy storage. Both processes can occur in aerobic and anaerobic cells alike. The aerobic part comes after glycolysis, which if it's anaerobic, it goes to fermentation, and if it's aerobic, it goes to electron transport chain. Glycolysis requires the cleavage of the fruit requires the aldolase enzyme to cleave the fructose biphosphate into 2,3 carbon units, which will become the pyruvate. And then aldolase, the aldolase enzyme itself has different isoforms, optimized for different body parts. Isoform A in the muscles, this gives you more immediate glycolysis where it's needed. Isoform B in the liver for gluconeogenesis, to take your non-carbohydrate molecules and store them as fat or be sent into the bloodstream. And isoform C in your brain for what can be hypothesized to be is meant for stressed hypoxic situations. And furthermore, the expression of aldolase is regulated by transcription factors, which are proteins that either block or promote RNA polymerase by binding to the DNA. All right. If you have any questions, please reach out to me. Here is a list of references I used. You can also use them for yourself for further reading. I hope this information finds you well, and the video link will be posted ASAP.